What we are looking at right now is a cross-section of cardiac muscle. Cardiac muscle tissue, also known as myocardium, is a structurally and functionally unique subtype of muscle tissue located in the heart that actually has characteristics from both skeletal and muscle tissues. It is capable of strong, continuous, and rhythmic contractions that are automatically generated. The contractility can be altered by the autonomic nervous system and hormones. In addition, this tissue type has high metabolic, energy, and vascular demands. Cardiac muscle fibers are long, branched cells shaped like cylinders, joined end to end, with one or two nuclei located centrally. The fibers are separated by collagenous tissue that supports the capillary network of cardiac tissue. The myofilaments of cardiac muscle are arranged in a similar pattern to skeletal muscle, resulting in cross striations. The fibers are crossed by linear bands called intercalated discs. These structures has two important roles. Firstly, they provide attachment points that provides the tissue with a characteristic branch pattern. Secondly, they allow cardiac muscle tissue to function as a functional syncytium. Essentially, the contractile stimuli is propagated from one cell to the next one, resulting in a synchronous contraction of the entire tissue section. While the majority of muscle cells within cardiac tissue are specifically contracting, there is a special set that performs another role. They are called cardiac conducting cells, and they automatically initiate and propagate the contraction impulses. Cardiomyocytes, also known as cardiac muscle cells, usually contain one elongated nucleus that lies in the center, which is a distinguishing feature from skeletal muscle. By examining the altered structure, it becomes apparent that the myofibrils separate as they approach the nucleus, pass around it, and reassemble in the original pattern on the other side. You can visualize the arrangement by imagining two cones that are joined at their vertices, which represents the nucleus. In fact, cell organelles are also concentrated in the cytoplasmic region around the nucleus. These include mitochondria, Golgi apparatus, lipofuscin-filled granules, and glycogen. Lipofuscin is a red-brown pigment, often called the wear and tear pigment, which gradually accumulates inside cardiac tissue with age. It is the remnant of lysosomal cell contents. The cytoplasm of cardiomyocytes, called sarcoplasm, is eosinophilic and appears as a 3D network. Due to the high energy requirements, cardiac muscle tissue contains additional large and elongated mitochondria located between the myofibrils. They can run the full length of the sarcomere and contain many internal cristae. In addition, extra glycogen granules are also located between the myofibrils to store the energy. Threads of collagenous tissue fibers together with capillaries are also present between the muscle fibers to provide the tissue with support and a blood supply. Now, talking about the intercalated disc, cardiac myocytes are joined together via intercalated discs, which coincide with Z-lines. They appear as lines that transverse the muscle fibers perpendicularly when examined with a light microscope. However, if the ultrastructure is examined, the discs are far from linear. because they have finger-like interdigitations to maximize the contact surface area. The disc also contains two compartments that are orientated transversely and laterally in relation to the myofibrils, resembling a flight of stairs. That is so awesome! To accomplish their attachment roles, intercalated discs contain three types of cell junctions, and let us look into this. Adherence junctions are the fascia adherents. They are a part of the transverse component and are the ones making the intercalated disc visible in hematoxylin and eosin staining. They are responsible for actually connecting the ends of the myocytes together to form a fiber. In addition, they transmit the force of contractions from cell to cell because the actin filaments of terminal sarcomeres insert into this junction. Second is desmosomes or the musculae adherentes. They are part of both components and they reinforce adherence junctions. They prevent the separation of myocytes during contractions by anchoring intermediate filaments. Third is the communication or the gap junctions. They are part of the lateral component of intercalated disc. They allow cardiac tissue to function as a syncytium by providing pathways for various ions to pass between adjacent cells, resulting in the propagation of excitation and subsequent contraction. What about myofibrils and sarcomeres? Sarcomeres are the functional subunits of myofibrils and the contractile units of cardiac muscle tissue. They are arranged into a branch pattern forming a 3D network in the cytoplasm. Sarcomeres are specific portions of myofibrils located between two Z lines and are responsible for the striated appearance of cardiac tissue. They are composed of thick and thin filament. Thick filaments are composed of polymerized myosin type 2 protein and are attached to a band called the M line that is situated in the middle of the sarcomere. Thin filaments consist of polymers of the protein alpha-actin and are attached to the Z lines. These two lines, together with the A band that corresponds to the length of the myosin filaments, are electron-rich and appear darker in electron microscopy. The I and H bands appear lighter and they represent regions which consist of only thin or thick filaments, respectively, but not both. The cytoplasmic regions between the sarcomere branches are filled with mitochondria and smooth endoplasmic reticulum called sarcoplasmic reticulum, which envelopes each myofibril. The membranous network of sarcoplasmic reticulum is traversed by structures called T-tubules, which are extensions of the sarcolemma or the plasma membrane of muscle cells. They form the T-tubule system, and their lumens are communicating directly with the extracellular space. Their course follows the Z lines of the sarcomeres, resulting in a single T-tubule for every sarcomere. The region formed by the pair of flat terminal cisternae of the sarcoplasmic reticulum and a portion of a T-tubule is called a triad. Looking at the cardiac conducting cells, remember that contractions are initiated and propagated throughout the heart by specialized cardiac cells called cardiac conducting cells. They are not neurons. Collectively, They form the conducting system of the heart. These cells form specific structures like nodes, bundles, and conducting fibers. The initial spontaneous stimulus starts from the sinoatrial node located in the wall of the right atrium at the level of the entry point of the superior vena cava. The impulses travel through the walls of the atria, resulting in contraction. They are then picked up by the atrioventricular or the AV node situated above the tricuspid valve in the medial wall of the right atrium. These two nodes are enveloped by collagenous tissue that is full of capillaries and autonomic nerves. After the AV node, the impulse passes through the bundle of His, the right and left bundle branches, and finally through the Purkinje system. The three bundles stain quite pale due to their high content of glycogen granules and mitochondria. The Purkinje fibers also contain a central area that stains pale. Cardiac conducting cells are connected strictly via desmosomes and gulf junctions only. They also don't have a T-tubule system. Well, as you can see, the contraction of the heart is spontaneous. However, despite their autonomy, conducting cells are not isolated from the nervous system. The sympathetic branch increases the impulse frequency from the nodes to the conducting system, while the parasympathetic branch decreases it. As every impulse is followed by contraction in normal situations, the rate of contraction is increased or decreased respectively. And what can we do? We need to study the contraction. We cannot complete studying the cardiac muscle tissue without looking into its contraction. The contraction mechanism is similar to that of skeletal muscle. Basically, the depolarization of the sarcoplasm travels through the system of T-tubules, all the way to the sarcoplasmic reticulum. Voltage-gated channels open and calcium ions are released in the sarcoplasm. These ions allow the myosin and actin filaments to form cross bridges and subsequently slide past each other, sliding filament mechanism. The excitation and contraction are passed on to the next myocytes via intercalated disc and cell-to-cell junctions. Despite the close similarities between cardiac and skeletal muscle tissue, there are several significant differences. Firstly, the depolarization of the sarcoplasm lasts longer in cardiac tissue. In addition, calcium channels are also present in the walls of the T-tubule system, rather than being limited strictly to the sarcoplasmic reticulum. In turn, the released calcium ions bind to calcium-sensitive channels in the sarcoplasmic reticulum, which results in a large and fast release of further calcium ions required for contraction. As to its clinical points, myocardial infarction. Okay, as we have read and heard previously, cardiac tissue requires a high and continuous supply of energy and oxygen. The heart's oxygen supply is brought to the heart via the coronary arteries, which are highly susceptible to atheromas. These are abnormal deposits of fatty acids, cholesterol, and various cell debris. If these atheromas keep increasing in size, they eventually occlude the coronary arteries, resulting in a reduction of oxygen supply to the tissue. This lack of oxygen leads to a condition called myocardial infarction, which represents the death of cardiac tissue. As part of a normal physiological response, the affected area is repaired and replaced with fibrous tissue that interrupts the propagation of the excitatory stimuli and subsequent contraction of the heart. Such asynchronous contractions can cause arrhythmias or disturbances of cardiac rhythm, an example being ventricular fibrillation. Another, cardiac hypertrophy. Cardiac hypertrophy means an increase in size of cardiomyocytes. To accommodate this large size, the cells need to assemble more sarcomeres and synthesize more mitochondria. They are also marked by enlarged nuclei and greater protein production. Hypertrophy has several possible causes, each one leading to a particular pattern or type. Pressure overload hypertrophy is often caused by hypertension. It involves an increase in the myocyte cross-sectional area due to the assembly of new sarcomeres in a parallel fashion to the old ones. It usually affects the ventricles by increasing the thickness of their walls. Second, volume overload hypertrophy is caused by an abnormally high blood return to the heart. It involves new sarcomeres being formed at the end of old ones, increasing their length of the myocyte rather than their thickness. This type dilates the ventricles and makes the heart heavier. Now, cardiac hypertrophy results in a heart that has very high metabolic and oxygen demands, but insufficient supply due to the extremely high consumption and lack of new capillaries. In turn, the workload is increased and ischemia is possible, eventually resulting in cardiac failure and death. Now, it is important to realize that cardiac hypertrophy is a completely different condition compared to myostatin-related muscle hypertrophy. The latter is rare and genetic in nature. In addition, it does not cause any medical problems for the affected individual. Cardiac muscle tissue. And that's what we have learned today. I hope my voice could help you, those who are already tired of reading their books. I'm sure you will learn a lot about cardiac muscle. Thank you so much for listening about this lecture on cardiac muscle. Please do not forget to like this video and share this to your friends, to students who really need this lecture. Kindly subscribe to my channel if you haven't subscribed yet. Thank you so much and have a nice day. See you again on my next uploaded video. Good day.